Good day, everybody. Thank you for attending this webinar. In this first module, I'm going to be focusing on a little bit of background on SciCon innovation, as well as the technology behind the SciFlow 1000 fluidic culture system, pictured here in the center of the slide. For additional information, if you want to contact us, please visit our website or email or telephone us directly. Thanks. I'm going to get started. A little bit about SciCon innovation, what we do. There are three parts of our business. There are three facets to our business. Historically, since about 2009, we've had a skin and tissue procurement business, and we also have a proprietary line of media that we sell called cryothaw, which is very good at freezing down cells, leaving the cells in a viable state so that when they're thawed out, they recover and function in a biologically relevant fashion. Additionally, we have CRO services. So we run projects for customers and we deliver data or results back to those customers. Predominantly, though not exclusively, the CRO services are focused at demonstrating the benefits and features of the SciFlow 1000 fluidic culture system. We also have customs tools business where we help people design novel fluidic systems as well. For the rest of the talk, I'm going to focus in on the SciFlow 1000 as that is the core business moving forward for SciCon innovation. What are some of the advantages of the SciFlow 1000? So there's three areas I'm going to focus in on. The first being biological relevance. I'm going to get into more information about SciFlow as the talk goes on, but SciFlow 1000 is a fluidic culture system that connects together multiple wells in series in a standard tissue culture plate. With those multiple connected wells, one of the things you're able to do is you can connect cells of different types together in series across the plate. So you could have upstream liver cells, you could have downstream target organ of interest, you could have downstream off-target cells as well for looking at a whole biological metabolism, toxicity, efficacy type of study. Additionally, because it is a fluidic system, we have gradients. Gradients are a key feature of many biological systems. We get gradients of compounds, gradients of metabolites, gradients of cellular responses. I'm going to go into a lot more detail on gradients in the applications portion of this talk. Compatibility. Because the SciFlow 1000 is an SBS compliant 96 well plate, we are compatible with standard plate readers, standard assays, standard high content imagers that are available on the market. So while it's a new system, there's no need to change your workflow. There's no need to change the technologies. There's no need to change the instrumentation and kits that you're already running in your lab. And last but not least, the system is very data rich. What I mean by data rich is you can generate a lot of information very quickly and efficiently from a few number of SciFlow 1000s. We do a lot of high content imaging. We do a lot of real-time analyses. So all the data shown here in this last panel comes from a few rows of one SciFlow 1000. So each plate tells a story. There's a lot of information you get from each of your experiments. So to summarize, the benefits are a biologically relevant system, usability and compatibility, and a data-centric system. To take a small step back from the technology, at the heart of the SciFlow 1000, it is a surface, it is a system for culturing cells. I'm now just going to show a small set of cells that we've cultured on the SciFlow 1000. On this slide, I'm highlighting it on human cells. SciCon has a long history with liver cells. Our founder has a long history with working with liver from multiple different species. What I'm showing on this slide are human hepatocytes at day 0, human hepatocytes on day 1, some non-parenchymal human cells, as well as some work we've done with breast cancer or MCF7 cells. There's many other human and non-human cell types we've also successfully cultured on SciFlow. I also want to mention that SciFlow 1000 is compatible with collagen coatings, it's compatible with Matrigel, it's compatible with fibronectin. There's a lot of different surfaces that you can put on SciFlow for culturing specific or alternative cell types. A lot of the work I'm going to talk about in the applications portion of this talk focuses on HEPA-RG cells. HEPA-RGs are a more biologically relevant cell line for looking at liver metabolism and liver safety and efficacy studies. What I'm showing here is a 24-day time course of culturing the HEPA-RG cells in the SciFlow 1000. I also want to mention all the images I'm showing are directly in the SciFlow 1000 and visualized with a standard inversion microscope. So there is no special instrumentation you need for looking at your cells, for checking on your cells, for grabbing quick pictures of your cells to make sure everything is progressing normally and the way you want it to move forward. I mentioned SciFlow is also compatible with high content imaging. I wanted to put in this image of a little bit of fluorescence imaging work that we did. These are some HepG2 cells that I'm showing that are stained with Herc stain, which is shown in blue, and Celltox green, which is shown in the pinkish reddish color, and it's just measuring cell viability directly in the SciFlow 1000 in a Molecular Devices Image Express high content imaging system. Because it is a fluidic system, we often get asked, are the cells healthier? Are the cells more metabolically competent? Do the cells look more biologically relevant? What I'm showing here on the top two panels are day one and day 14 images of the HEPA-RGs in the SciFlow system. On the bottom, I'm showing again day 1 and day 14 of those same HEPA-RG cells cultured in static plates. Visually, they look very similar. The cells in SciFlow, the cells in static, they have a similar morphology, similar cell phenotype as we're looking with our standard inversion microscope. We also measured cell titer glow, or ATP, on day 14 in both the static and the SciFlow systems, and we can see relatively similar amounts of ATP in both the SciFlow and static systems on day 14. We also looked at GSH. GSH is a measure of reduction potential. We did this with the Promega kit GSH Glow, and we can see that there's a roughly 50% higher level of GSH in our SciFlow, in our cells cultured in our SciFlow plates than the cells cultured in static plate. So the cells do appear healthier, they do appear a touch more metabolically competent, or at least phase 2 metabolically more competent in the SciFlow versus the static system. I now want to focus on the SciFlow technology. How does the system work? And then I'll get into some of the applications or benefits of the SciFlow system in the second module. When people first get a SciFlow 1000 in their hands, they sometimes tell us that it looks complex. It looks like there's a lot going on. Well, one of the things that SciCon has done is we've engineered simplicity into the system. There is a lot of CAD work behind the scenes. There's a lot of fluid mechanics. There's a lot of material science that went into developing the SciFlow 1000. But the end result is a very user-friendly solution which is ready to use right out of the box. It comes pre-packaged, individually sterilized, tissue culture treated, and ready for you to culture cells directly in the SciFlow, directly in the system. SciCon has handled the engineering so our customers can focus on the biology. I also want to mention, not pictured here, the SciFlow 1000 does come with a lid. It comes with a lid to maintain sterility and allow you to move the plate around the lab without opening it up and exposing it. I just didn't have the lid in this picture because it made it more challenging to see the system. I've mentioned multiple times that the SciFlow 1000 is a fluidic culture system. So the question is, what drives the flow? What causes the flow from well to well across the plate? What I'm showing in this image is a side-on view of a SciFlow 1000 with a blue indicator dye in one row of the plate. What I'm highlighting here is the 0.5 millimeter or 500 micron decrease in well height as you move from left to right across the plate. It's this decrease in well height that drives the fluid flow. It's this hydrodynamic pressure that causes the SciFlow 1000 to flow from left to right across the plate. All of this flow is accomplished with no pumps or no tubing. The SciFlow 1000 is made entirely out of one material. It's made entirely out of tissue culture treated polystyrene. And as I said before, but it's worth repeating, it is an SBS compliant 96 well footprint. So it works with your standard plate readers, high content imagers, multi-channel pipette man, and laboratory robotics that you may already have in your lab. Gradients. I mentioned gradients on an early slide. This is a top-down view of one row of a SciFlow 1000. Not shown all the way on the right side of the plate is a well or a sink or an overflow well in column 12. But I want to highlight column number 1 or well number 1, which is the dosing well or the source well. This is a larger well. It accommodates approximately 500 microliters of media, and it allows for uninterrupted culture for longer periods of time. So what you see across your SciFlow 1000 is you get a source well on the left, a sink on the right, and then 10 identical culture wells in the middle of the plate. There's 10 identical wells for culturing cells across the SciFlow 1000. Each of these wells is the same size as a half area 96 well plate. When you're using the system, you're adding compound or drug into the source well, and the fluidics causes that to flow across the plate from left to right. What this does is this generates a gradient. You get a gradient of test compound. As the test compound flows across the plate, it dilutes into each well of the plate and then continues flowing downstream or downhill. So you get a decreasing concentration of your test compound from left to right across the row. Additionally, if your cells are able to break down that test compound, you get metabolites generated. Those metabolites also flow across the plate, but what you get is you get an inverted gradient of metabolites. As the metabolites are being generated uphill or upstream, but continue to flow across the plate, you get accumulation of those metabolites in the downstream wells. So essentially what we get is we get a separation. We get higher concentrations of test compound upstream and higher concentrations of metabolites downstream. So where we see a certain effect, in what wells of the plate we see a certain effect, give us information about what is causing that effect. If we see cell death, if we see efficacy of our compound in the upstream wells, we know it's a compound-mediated effect. If we see our effects in the downstream wells, we know it's more of a metabolite-mediated effect. So there's a lot of mechanistic information we get from SciFlow in addition to just the compound itself and the safety and efficacy of that compound. Exposure and fluidic performance. We frequently get asked, how does SciFlow work? What do the exposures look like? How does the compound flow across the plate? So what I'm showing here are two different schools of thought, two different ways of dosing cells with compounds or drugs. On the right should be very familiar. It's a static system. When you add a drug or compound, that drug or compound immediately goes from 0 to 100% and then sits at that same concentration for the entire study. That's ignoring cellular metabolism, but cellular metabolism is usually a small percentage of the overall drug concentration that you're looking at. If you want to investigate different drug concentrations, you can manually set up your own dilutions, and you can do 2-fold or 3-fold dilutions across the plate to look at various drug concentrations, each within its own well. It's a fixed concentration, and it's a known concentration. Contrasting that with SciFlow and its fluidic exposures, what I'm showing here is a time course across multiple days in multiple wells within a SciFlow that are all connected. What you see with the blue line, or a very upstream well, is there's a rapid increase in compound concentration across a few days, and then it reaches an equilibrium where it sits for the rest of the study. In well 7, or somewhere in the middle of the plate, it's a much more linear exposure that we see, as it takes time for the compound to flow across the plate from the source well until it does finally accumulate way downstream, or days 7 or 8 later in the experiment. Last but not least is a well all the way at the right side, or the downhill end of the plate. There's a longer lag phase and then a slower increase in drug or compound concentration in those downstream wells. So what we're seeing are very different, very unique dynamic concentration gradients over time. I do want to stress that the concentration gradient is tunable by the end user. How much volume you add into the source well and how frequently you add more compound into the source well affect these gradients, affect the way the flow goes, and affect the exposure of the compound. So you can expose cells to higher or lower concentrations as may be dependent based on the particular experiment. We frequently get asked, how fast does it flow? How far does the compound flow? How many doses do I have to give so my cells see my compound of interest? Shown on the top panel is a picture of a SciFlow 1000, again top down, and we're adding a blue dye into the source well just to visually show you how the dye is flowing across the plate, diluting across the plate over time as it flows from the source well downhill into the sink or waste well all the way on the right side. In the lower panel in the graph, I'm showing a fluorescein titration experiment, or a fluorescein flow experiment. It's a 60-minute flow, and what I'm showing is upstream wells and then more downstream wells, so you can get a sense of how the flow goes across the plate. What we generally see, or what we always see, is there's a quick burst phase where it takes about 15 minutes of rapid flow, where there's a rapid increase in drug or compound concentration in the upstream wells, and that 15 minutes is followed by a much slower flow, which persists for about 90 minutes to 2 hours, where there's a more gradual increase as the compound is slowly spreading across the plate. Beyond about 2 hours, there is a slow diffusion-limited process that's driven across the plate based by evaporation and some of the, there's a wick in the waste well. The wick also helps to draw a little bit of material across the plate, but it's a much slower diffusion-limited process. So the flow itself lasts for about two-ish hours in a somewhat rapid format, and then it's much slower and will sit there until a user intervenes, until a user adds more compound, adds more media into the source well to keep the flow going. I do want to stress, though, that the equilibrium volumes in all of the wells always keeps media in all of the wells, always keeps the cells covered with media. It does not dry out, so you do not need to intervene with the system every few hours. You can easily go once a day, twice a day, even 3 times, once a day, every other day, every third day, if required by the experiment. The last slide for this module that I want to talk about is some of the applications that we've done on the SciFlow 1000. As I mentioned before, we have a long history with liver, so there's a couple of different applications with liver or injury or DILI that we looked at. We looked at a study with aflatoxin. We also did a study with acetaminophen, which I'll go through in the applications portion of this talk. There's also some work we did with multi-organ tissue systems where we had liver cells upstream of MCF7 or human breast cancer cells, and we were looking at bioactivation of a panel of chemotherapeutic agents to see if we could capture the metabolism of those drugs and monitor the efficacy of those drugs based on metabolism. Thank you for your attention. If you want to learn more about these applications, there's an applications module for you to view. Thanks.